Plants grow and reproduce themselves in every kind of environment on Earth. To be able to survive, they have to be able to receive information about the world around them. And they must be able to respond to these signals. If you put a plant on a windowsill, you'd expect it to bend towards the light. And if you plant a fruit or a seed, you expect the root to grow down and the shoot to grow up. And we all expect that given the right conditions, fruit will ripen and in most cases change colour in doing so. But what are the internal control processes within the plant that coordinate these events? What growth regulators are involved? Surprisingly, little is known about such physiological mechanisms. In this programme, we'll investigate just two responses: the growth of stems towards a source of light, called phototropism, and the control of ripening in fruit. Let's start by taking a closer look at phototropism using time-lapse filming. As the light source is moved from side to side of these cress seedlings, the shoots grow from one direction to another towards the stimulus, and the plant starts to bend within 10 minutes. Over 100 years ago, in this house in Kent, Charles Darwin was fascinated by his study of plant growth in varying light conditions and performed some simple, now classic experiments. He grew oat seedlings and observed what happened to the coleoptiles. He covered the tips of some of the seedlings with tiny metal caps. He found the cap seedlings didn't bend when illuminated from one side. And he noticed too that the untouched plants bent not at the tip, but farther down the shoot. Darwin likened the tip to a primitive brain, sensing the light and then sending some influence down the shoot. About 40 years after Darwin made his observations, other workers, particularly Went, tried a more physiological approach and thought they'd solved the puzzle and understood what was going on. Darwin had established that there must be some kind of link between the tip of the shoot and the bending region, but they wanted to discover the nature of the link. Was it chemical or physical? In one of their experiments, the tip of a shoot was removed and placed on agar and illuminated from one side. Any chemical produced would diffuse into the agar. The agar was then replaced on the cut shoot together with the tip. In a similar experiment, a piece of mica was inserted between the agar and the cut shoot. Both plants were then evenly illuminated. Each one bent towards the direction of the original light source, which stimulated the cut tip. Chemicals can't diffuse through mica, and the bending movement of this plant was reduced. The researchers postulated a neat theory to account for these movements. They thought that some plant growth substance in the shoot tip is activated by light and passes down the stem to cause elongation in the cells behind the stem tip. This growth promoter, or auxin as it was called, only moved down the shaded part of the stem, and so this grew more quickly than the side receiving the light stimulus, and bending towards the light occurred. This became known as the Kolodny-Went theory. For an overview of some of this work, we visited York University and spoke to Richard Fern.
Went was working in Holland in the late 1920s, trying to understand coleoptile phototropism, where the view was that the apex was in fact important. Kolodny was a Russian, and he was working on root gravitropism largely, but he also had the view that the root apex was important in controlling root growth. And the two workers proposed independently that substances coming from the apex might be distributed unequally when there was a case of gravitropism or phototropism, and that would explain the unequal growth which bent the organ.
But more recently, Fern and other workers have questioned these basic ideas and assumptions. To analyze the growth movements of plants during phototropism more accurately, we can use a very simple technique. Tiny beads made sticky with lanolin are placed at intervals in rows on opposite sides of a seedling shoot. To observe what happens, a video image analyzer can be used. A video camera is carefully positioned to focus on the seedling and is connected to a monitor screen of the image analyzer. The apparatus carefully plots the distance between any two particular beads and records the data. This is repeated for every space between the beads on the shoot. The plant is then illuminated from one side only. After an interval of time, the measurements will be repeated. A typical result of such an experiment is seen here. The graphs show what happens to the shaded part of the shoot, shown in blue, and the illuminated side, shown in yellow. When unilateral light is applied, there's a short time lag while the cells start to respond. Then the cells of the illuminated side of the shoot stop growing completely, whereas the ones on the shaded side keep growing at approximately the same rate as before. What can we deduce from this? Well, it suggests that the bending movement is caused not by the action of a growth promoter or auxin, promoting cell growth on the shaded side as predicted by the Kolodny-Wendt theory, but by the action of a growth inhibitor on the illuminated side. Does this mean that their theory is wrong?
Well, there are two recent observations which definitely contradict the traditional view. The first one is that if you actually measure the amount of auxin in the shaded and the illuminated side of a dicotyledonous plant as it undergoes phototropism, you don't find any change in the amount of auxin in the two sides of the plant. That contradicts the predictions of the Kolodny-Wendt theory. Even if you look at the cells which are controlling growth, which are only a subpopulation of the total cells in the organ, you don't find the changes, which was one last hope of the theory had, that you would find the changes. And then the other bit of information which has contradicted the theory is that someone's actually repeated Wendt's original measurements of auxin coming out of a tip which has been subjected to phototropic stimulation. And they find, in fact, that there is no difference in the amount of auxin coming from the shaded side or the illuminated side of the tip, which again contradicts Wendt's original idea. There are two contradictory views as to what might be happening. One is that people still believe auxin might be causing the response, and they argue that it must be, the changes in auxin concentration must be occurring at a subcellular level. And the second alternative is that light directly inhibits cell elongation, either by changing the level of some other substance in the cell, such as an inhibitor, or by having a direct effect on cell enlargement, and not involving any particular hormone at all.
There were few ways of studying what was actually going on inside the plant tissue when Darwin and later Wendt and Kolodny were postulating their ideas on plant growth. Modern techniques enable minute amounts of chemicals produced by plants to be isolated, identified and quantified. High-pressure liquid chromatography is one such technique. The plant extracts are sampled automatically by mixing with various chemicals, and then the individual components of the mixture are separated out by passing them through a column. When they come off the column, they're passed through an instrument which will detect the individual parts of the mixture. The monitor screen displays the information. The time that the compound has spent on the column tells you what the compound is, and the size of the peak tells you how much of the substance is present. Radioimmunoassay is another technique frequently used to analyze compounds produced by plants. In the radioimmunoassay, you have the advantage that you're using an antibody against the plant growth substance to identify it, and therefore you don't have to purify the mixture very much, because the antibody, in theory, would only recognize the compound you're interested in. So, instead of identifying a lot of compounds, As in high-pressure liquid chromatography, it will only analyze the one compound you're interested in. You essentially measure how much antibody there is by measuring how much of a product is released by the enzyme attached to the antibody. In this case, it releases a yellow dye, so you see more yellow dye when there's more of the antibody present. So, what do we now know? We know that the presence of auxin and its quantity in an extract from any part of the plant can be qualitatively measured using high-pressure liquid chromatography techniques and radioimmunoassay. We also know that bending towards a light occurs somewhere behind the tip of a shoot. And although there's more response when the tip of a shoot is illuminated, there's still some bending response, even though the tip has been completely covered. So let's consider what Darwin actually said about his observations.
Darwin tried to show that the apex was important by covering the apex with a small opaque cap, and he reasoned that if the apex was a site of light perception, that if you covered it, the plant wouldn't show a phototropic response. And when he did that experiment, he did in fact show that that was the case under certain circumstances. He noted that it wasn't always true, but it could be true. Most modern textbooks only quote a few lines of Darwin's volumes which he wrote on this, and they picked up on his work that coleoptiles required their tips to be illuminated in order to show phototropism, and they assumed that that was a general feature, whereas even Darwin said that, for instance, oat coleoptiles, that was not the case, that if you illuminated the lower regions, they would show a phototropic response. So they picked up one example of his work and said this was a basic general model for how shoots control their growth, which it doesn't seem to be.
So if Darwin's observations and Kolodny and Wendt's theories are now in doubt, what possible mechanisms could be involved in controlling the phototropic response?
An important point to be remembered when we're talking about phototropism is that in the end, the bending is dictated by differential growth between the upper and lower surfaces. Now, differential growth is regulated by all sorts of changes, changes in the cell wall, changes in the cytoplasmic volume. And these are the result of probably regulation ultimately of different genes. Now, if we have different genes that are being turned on, maybe in the lower side in the gravitropic organ, or turned on the side that is away from light in a phototropic organ, then we have an opportunity there of trying to unravel which genes are turned on and which genes are turned off using the molecular biological techniques that are now available. We are now in a position where we may be able to attempt to identify messenger RNAs that are associated with growth. If we can identify these, then we have an opportunity of trying to ascertain how these particular genes which code for these messages are actually turned on.
However auxin is involved in phototropism, it's certainly an important plant growth regulator. Another such substance which is involved in many physiological processes in plants is ethylene. One particular interest in ethylene at the moment is its effect in controlling the ripening of fruit, particularly tomatoes. Both in Europe and the USA, tomatoes are big business. Vast quantities are used just to go round baked beans, let alone the huge numbers that we consume in salads. The commercial production of tomatoes has reached a high level of sophistication, with thousands of acres being grown under glass. They're grown in an artificial medium, which is automatically monitored and supplied with water and nutrients. Even pollination gets a helping hand. In this nursery, insect pests are reduced by biological control methods. The eggs of predator organisms are hatched out to rid the glasshouses of pests. Because they're grown in near perfect conditions, these plants grow to about 20 foot in height, and to aid the picking of the ripe fruit, the plants are supported by strong thread. This is played out as the plants are lowered and the ripe fruit brought within reach. Tomatoes come in every shape and size, and if you can grow your own and eat them within a few hours, then great. They'll probably taste terrific. A lot of money has been spent in the industry devising a production and distribution system which is geared to rapid delivery to the point of sale. But despite every effort, we all know those grown at home tend to taste better. But why? Let's consider what happens to them before they get to us. After the tomatoes are washed, colour graded and packed, refrigerated lorries take them to the sales outlets, and then they're usually stored in fridges at home. But they only remain perfect for 4 days, and by the time we get them, they're frequently tasteless, overripe and squashy. Processing factories, wholesalers and the public all have different requirements, but everyone wants a tomato with a good taste and a longer shelf life. Just as it's turning from green to red, the chemicals inside it are perfect for taste. So, what we need is a tomato which ripens much more slowly and doesn't need to be refrigerated. A possible answer was found in the South American jungle, the tomato which stays green. But sadly, it's inedible. At the Institute of Horticultural Research, an experiment is being conducted to cross this tomato with the sweet-tasting, short-lasting Ailsa Craig. A perfect tomato is produced from this cross, which might last for 2 months. But Ailsa Craig has such a low yield that it's unsuitable for commercial growers, so it's being used by plant physiologists to develop a system of isolating and exchanging certain genes to produce a genetically engineered or transgenic tomato. And the technique perfected here could also be used on commercial varieties, since mutant strains exist for many of them. Although 86 substances are involved in ripening, at present scientists are only working on the genes responsible for softening of the tomatoes. Softening is brought about by ethylene, causing the production of the enzyme polygalacturonase. This substance weakens the cell walls, and the result is softening of the fruit. The binding of ethylene to a receptor is the first step in a sequence of events that produces new ripening-related proteins, such as polygalacturonase. This must occur by turning on a gene located on a section of DNA. On the DNA, part of one strand of the double helix carries the coding for the protein polygalacturonase. This strand is called the sense. The other strand contains the mirror image and is called the antisense. Messenger RNA is produced from the template of the sense DNA strand. The messenger RNA passes out of the nucleus to the ribosomes in the cytoplasm of the cell, which are the sites of protein synthesis. The individual amino acids are then linked together in the right order to produce, in this case, polygalacturonase, or PG as it's frequently called. Professor Don Grierson and his team at Nottingham University have now managed to isolate the gene which encodes for the enzyme for PG, and using DNA splicing techniques, they've swapped the sense and the antisense strands on that part of the DNA. This means that messenger RNA for PG can no longer be produced. So, instead of the gene for PG being read, nonsense messenger RNA will be produced. The result is that no PG can be produced by the cell, but all the other enzymes associated with ripening, affecting taste and colour, will still be produced normally. In the laboratories of the Institute of Horticultural Research, the transgenic tomatoes are being tested for firmness and colour. During firmness testing, the tomatoes are compressed and the readings are recorded by a computer. But constant, predictable colour of the correct shade is vital when selecting a variety for commercial development, since the marketplace dictates predictability and uniformity. So, colour testing is also carried out. Because these anti-sense tomatoes don't go squashy, it should be possible to leave them on the plant long enough for them to develop full flavour, be ready to eat, and yet still have a long shelf life. The final evaluation of Professor Grierson's project is being carried out here. The results are encouraging. The yield of the anti-sense plants is about the same as ordinary Ailsa Craig, and there are only very slight physical differences. The anti-sense technology employed to produce these long-life tomatoes can also be used to study other physiological processes within plants, such as phototropism that we considered earlier.
The anti-sense technology is a very sophisticated and elegant way of turning off a particular gene product. By inserting, by genetic engineering techniques, an anti-sense message into a plant, then that genetically engineered plant, or transgenic plant as it is called, can have particular genes turned off at particular stages in its development. For example, if we could identify a gene that was a putative gene coding for a message associated with growth that was turned on a shaded side of a coleoptile or a cotyledon, then we have the opportunity to turn it off specifically by anti-sense technology. If we turn it off, we will make certain predictions. That is, that the coleoptile or the cotyledon might not respond to light. Having identified particular genes, we then are able to work back from that position. So, starting with light, light is transduced into various different second messengers which ultimately may turn on specific genes. We can work back from that and maybe identify, if a hormonal signal is involved, how that hormonal signal actually interacts with the specific genes that we know associated with the phototropic response.
Could anti-sense RNA technology, do you think, have commercial implications in the future?
If we can turn off genes which, for example, may be associated with a detrimental process, perhaps senescence of an ornamental plant or some vegetable, then we might be able to maintain the shelf life of that particular plant. Similarly, if we could turn off some other product which is associated with some quality that we would prefer not to have, it might be that the particular plant material was too bitter, or it might be it had a certain aroma which was unpleasant, then again we could actually turn that off.
Because so little is known for certain about the physiological control exerted on the plant by plant growth regulators, and there's so much commercial potential there, it's an area of rapidly expanding research.
Some of the work that has been done already could well become commercially available within a few years. A lot will depend not on the nature of the experimentation and how fast that goes, but how quickly people are willing to accept transformed and genetically engineered material.
Whatever the arguments, in the future we should be able to understand and control the physiology of plant growth by a thorough knowledge of the role of plant growth regulators.